Okay, I think it's about time and we can get started. So good morning, everyone, and welcome to today's DASHU Virtual Journal Club. My name is Maisie and I'm your host today. And today we have a very exciting talk featuring Dr. Tai Xie and Professor Zhou Shi. Before I hand the mic over to our speaker, I would like to first spend a few minutes to give a brief introduction of DASHU and give you a heads up for our upcoming events. So DASHU is a 501c3 nonprofit organization providing a virtual community for people who are interested in statistics and data science to exchange ideas and help each other. We are a young organization founded about 6 years ago with a free membership. So far, we've had thousands of active members around the world. We host many types of events. We have hosted international scientific conferences, monthly virtual journal club like this one, and we collaborate with industry and academia to host career development events. We also have educational lectures such as the legal series. Please sign up for our membership if you haven't already to receive notification and participate in our conference and free events in the future. In addition to our official website, we also share our news and upcoming event promotions on social media. So we have a LinkedIn, Twitter, and a WeChat official page. We also post our video recordings of our post events on DASHU YouTube official channel. Please feel free to check it out. I would also like to advertise for our virtual journal club, which is a monthly event featuring high impact researchers presenting their exciting new work. Let us know if you would like to participate by presenting your research at our monthly virtual journal club. We always welcome speakers, topics, collaborators, as well as sponsors for our upcoming events. And as I said, we are a nonprofit organization run entirely by volunteers. We always actively seeking new volunteers. So please feel free to join us if you are interested. You can find us at dashu.org or scan the WeChat QR code to sign up and become a volunteer. Next month webinar, we have Dr. Bangard from Samsung Artificial Intelligence. He will give us a talk on the topic of automated image labeling for medical imaging AI. Please stay tuned and sign up for our membership to receive notifications about this event. And aside from the Virtual Journal Club, we have another exciting event coming up next month. It's the 4th Aung Symposium. It's a unique conference that brings statisticians and oncologists together to discuss the challenges in oncology drug development. This year's events will be hosted online. It's a 3-day event. Please feel free to register if you are interested in attending this Aung Symposium. Without further ado, it's my pleasure to introduce to you our speaker today. The first speaker is Dr. Tai Xie. Dr. Tai Xie is the founder and CEO of 2 companies, Brightech International and CIMS Global. He has over 26 years of clinical trial experience in pharmaceutical and CRO business. He is an expert in clinical trial operation, data management, e-clinical solutions, and statistical analysis. He is one of the authors who drafted the guidance for EDC clinical trials in China. Besides an entrepreneur, Dr. Xie is also an active researcher. He is a member of ESA, DIA, ASCO, and SAPA, and he is an active reviewer for various statistical journals. Our second speaker today is Professor Zhou Shi. Professor Shi is Professor Emeritus of Biostatistics and the former chair of Biostatistics Department and Director of Biometrics Division of the Cancer Institute of New Jersey at Rutgers University. Prior to joining academia, he spent over 17 years at Merck Research Lab. He also served in the US FDA Advisory Board for reviewing new drug applications. He is an distinguished statistician and opinion leader in both industry and academia. He has published over 116 papers in statistical methodology and medical research. His book, Statistical Design and Analysis of Clinical Trials, is a very popular graduate level textbook for many universities. He is also an ASA fellow and elected member of the International Student Statistical Association. So in today's talk, Dr. Xie will lead the presentation and Professor Shi will chair the Q&A sessions. And in this talk, they will introduce the concept of dynamic data monitoring for ongoing clinical trials and will demonstrate how to use this DDM technology in guiding a promising trial to success or detecting a hopeless trial to stop. So during the talk, please keep your devices muted. And if you have any questions, please feel free to type them in the chat box. And also when you are typing in your questions, we also ask the attendees to briefly state your background or professions with 1 or 2 words. For example, I am a graduate student in biostatistics, so I will just write graduate students in biostatistics along with my questions. And Professor Shi will select the questions and I will read it to Dr. Xie towards the end of our sessions. So now I will hand it over to Dr. Xie.
Okay, thank you. Yeah, can you hear me okay?
Yes.
Okay, great. Thank you so much for the introduction. And also I want to thank Da Shu for the invitation for this talk. I'm an entrepreneur for the past 16 years and also work in the industry as an employee for 10 years, also academic for 3 years. So my talk, you may have some feeling about the more kind of application than theory part. So the whole purpose is to tell you our idea, what is this dynamic data monitoring we are talking about. Now to tell the whole story, I just want to start with what is the situation in the current drug development. Okay, the first one is the survey actually in 2016, and they have some updated data, but number are very close enough. One thing I just want to point out here is about 70% of failure rate from phase 2 study to phase 3 study. In this survey report, they list a bunch of reasons, including manufacturer, chemical compound formulation, and a new drug come out of the market, increase the competition. But none of the reason is regarding the study design, which is the one we are going to talk about this because I'm a statistician, so I'm thinking about what is the, except those kind of extrinsic reason, what is the intrinsic reason we can talk about? That is whole.
Sorry to interrupt. Dr. Xie, can you share your screen again? I think there's.
Oh, really? Okay. I'm sorry.
Yeah.
Oh, because you are okay.
Yeah,
okay.
I shared my
You
slides,
share, okay.
So I cut it off.
Okay.
Yeah,
so,
sorry.
Sorry about that. Okay.
Yeah, now I can see the slides.
Okay, great. Sorry about that. I just directly jumped to the slides because the Meizi was here, her screen right now. I'm here. Okay, make sure you can see my screen and hear what I'm talking about. Okay. No, the story. Let me just repeat one more. This is the data in 2016. The point here is 70%, almost 70% of failure rate from phase 2 phase 3. Bunch of other reasons you can name, but one of the reasons is the study design. This is what as a statistician, we're thinking about. Now, to kind of like find the reason or understand what's the issue here, let's go back to the study design, very basic fundamental stuff. The very simple design, let me close this. Sometimes this, okay, I'm gonna move down. It's a traditional fixed sample design. What is the fixed sample design is you have estimate or assume the treatment effect, then estimate how many subjects you need for that study. For instance, you need 200 patients for that study. Now, fixed sample design, you don't take a look at the trial until they finished. Now, there's a two extreme situation to illustrate the idea here. Now, the first one is the one trial of the black color here put. It's trending up. It's a promise, but short enough, not good enough. Okay, the dotted line here, properly here is just a 1.96. It's for significance. Another red color trial is trending down. It's hopeless. Actually, we don't know until we unblind the study and do the analysis. We realize the first one, sorry, you have to do another one. If we know that, we may also do some sample size re-estimate, make that study eventually successful. But the red one, if we know earlier, we can terminate that study earlier, right? Save the money. And also on an ethical reason, if you continue a non-beneficial trial or drug is not ethical. So that's another reason. Now, our statistician, we also develop some other method. You can take a look at the data in the middle way. This is also we call, let me just try to see. Okay, it's a group sequential. Group sequential, you can take a look the data in between. Pre-specify where you want to take a look. And also who is going to take a look. And sometimes we have the double-blind randomized study. You need IDMC, Independent Data Monitoring Committee, take a look at that. And also for prepare this, it take about 3 to 6 months. As if you are in the industry, you know how long it will take. Programming, data cleaning, and also generate the output. And also you need an independent statistician. Now, traditional group sequential is just take a look at the data. Say you cross the upper boundary, you stop the study, or you touch the futility, you stop the study for futility. There's no learning here. Okay, we are talking about learning. You understand, right? We are talking about very fancy word called machine learning. There's no learning here. Now the next trial, or next design, which FDA also released a new guidance regarding the adaptive group sequential design. This one, as maybe you know, there's a learning here based on what you observe. You're going to reassess your assumption. Maybe you need more sample size. Then maybe you need to stop the study earlier. Or adaptation means you can do some justification, right? Adapt based on what you observed. Now again, there's some also issue here. One is you need a pre-planned where you want to take a look. This is number one. Number two, you still need to prepare that for IDMC. That takes some time, logistic issue. Three, I mean, still like independent statistician must be involved in this. There's a logistic ratio, but the key thing here, where you take a look, where you hold down interim analysis is the key here. So timing is an issue. Now, to also make this story more understandable, take a look. This is simulated data. I simulated 2 studies, just the example I showed before. Simulated data, one is a promising one, the black, and another one is not so promising, maybe hopeless. It's a fluctuate, just like you see the stock market today. You could jump down, jump up, and goes up. Now, where you want to join the market, it's very critical. If you jump around spots, you may make a wrong decision. You buy the wrong stock. It's the same idea here. So where you want to make your analysis, take a look at that. You may make a wrong decision. That's what I want to say the message for these slides. Now, another question is a nonlinearity. We always evaluate these things. We always assume what the data we deal with is a perfect normal distributed or identical IID situation. Now, as you know, all the data we are based on using the central limiting theory. Okay, there's a assumption for the one you can use that theory is identical independent distributed. This also led to other development, other theory for the group sequential, adaptive group sequential based on a so-called Brownian motion. The condition here requires independent identical distribution. This may not always hold, such as if the patient intracardiac change, okay, you may not have that kind of identical patient as you originally planned. And there's some other reason. You can name many, many reasons which may not be IID condition. For that kind of situation, you will see a nonlinear trend. We put it as a p-value. If you read something, you know what's the p-value. It's correspond to z-value, just times the square root of information fraction. Okay? So by times that square root of the information fraction, you linearize that. So in your p-value, you will see a linear trend. They're linear. But here you can see this data I just to illustrate the idea. You can see this may not be a linear perfect. Now, how to handle this? So by getting into what I want to talk to, I just want to list all those kind of situations. Now you may ask me, if while I'm doing this, I don't know, I mean, what is going to consequences? What's going to be hurt me? Here is extreme situation. Now, I give you two examples, extreme example. If you assume very optimistic, I mean, think that your drug is super miracle drug, you think your drug is 40%. It is 40% of your treatment effect. Assume the delta is the theta is 4%, but your true theta is 20%. It's half of that. Okay. Now, when you do sample size, you assume that because you assume 40%, you only need 133 because you are very optimistic. Now, if you want to do that, and the traditional way we always do, and half time we do the sample size re-estimate at 67, which is too early based on your true situation. Because in the true situation, 20% treatment differences, you may need the 526. The 67 is too early to do that. As you can see, the simulated data I showed you is very fluctuating. Now, a second extreme situation is the opposite way. If you are assume as 20%, you very prismatically, but your true drug is true treatment effect is much better than you expected. Your opposite way is too late also. So the whole thing here I put here is just to illustrate what is the question, what is the situation we are facing here. One is snapshots of data, timing. Another one, the decision DMC member gave to you is go or no go. They don't give you other things, maybe see or some other inclusion criteria, but whether your data, the trending situation goes up or goes down, or this is hopeful or hopeless, they are not give to you. Now, you probably already feel what I'm talking about, the issue, and also what it could be if we can change that situation, right? Continuous availability, data availability continuously, and automatically, because I mean, the technology currently is already advanced. We are so behind for the top technology. All those automation, even the car, you see the Tesla recently has some accident, but you trust. When we have the rocket on this space, we can recycle. You probably will think about the future clinical trial is possible for have a more continuity and automation. Now, so how can we do this? Okay, the technology I mentioned here is current clinical trial, 90%. I can say 99% of a clinical trial is managed by an electronic data system. It's called EDC. This is patient data. And all the treatment assignment is by our IRT system. It's sometimes called IWRS. This is for randomization. Compared to 20 years ago, you use envelope. We are no longer use envelope. Right now, machines doing this. Now, by combining these two machines together, EDC manages the patient data, IRT manages randomization, and also drug supply. So the machine will know who is taking which drug. So machine will unblind for you, no human handle here. So the data, you can combine these two and extract the data out. Machine will do the calculation for treatment differences for you. No human involved. So by doing this way, you can do many things. You can see which population may be more promise. What is the sample size you need? Maybe at the beginning, you expected estimate may not be enough. Many things you can also do it. Now, by summarize what idea I'm needs here, you can see the advantage. The same situation I put it before, if we continue to see, you will make this different kind of recommendation if you are in the IDMC member. Now, by summarize the idea, I'm going to move this. I don't know how to move this. Yeah, let's try close this. Sorry. Okay. Let me just say this. Okay. We are developing a system called dynamic data monitoring ecosystem. Why we call eco? Because we have patient data in the EDC. We also have the IWRS for randomization. These two systems will provide unblinded data. And we also are programming before in the system all those calculation for the stats like stopping boundary or sample size ratio and continue power everything as the patient get into. So we put that into other system called DDM engine to calculate continuously. Whenever new data, the patient reach the endpoint, the DDM engine will calculate that. We are also doing a simulation, use simultaneously parallel computing to calculate what you observed to ensure or create some confidence. We can repeat your study 10,000 times. So that's why we are putting into other module outside, because we don't want to slow down the ongoing clinical trial. Now, after we calculate this, we are going to display data in here. And this we call trial readout system. I mean, later on, I'm going to explain to you why we call the readout system, because we borrow this name from aviation industry. Now, there are 4 regions here. Now, first region is green. And what is the green means? We generate the green region by the condition power. For the target condition power, for instance, you want a 90% condition power. The above this region, if your data in this region, the condition power is guaranteed on your target condition power. Now, in the purple one, the region is less than the condition power you want. The lower bound is what? The lower bound is based on your affordability. Affordability is how for your study, how much like money you want, your sample size. Money is come back to sample size. You want double or you want triple. So when you have that number in your mind, you can calculate the data is lower bound. The lower bound, like this data, if you want to sample size estimate, that's okay. This data show you this study may be not good enough. But if you want to continue, you may also pay more effort, because maybe this is beyond your budget. Now, yellow region is, as you can see, the condition power is low and also beyond your budget, right? Now, the purple rose region here, it's conditional power based on alternative. It is already very low. The bar can be adjusted based on what you wanted to declare your study as a futility. So these 4 regions, when the data goes to this region, you can tell how your trial is, the situation. And also the line here is cumulative treatment effect. We plot on this readout system. So this whole thing, why we call it ecosystem, is all it's closed system. So data is come from patient or come from a real world data, or a variable device or a real world data. Now, let's move to next slides. Now, whole idea, I just want to say the whole idea is more like like a GPS. Everybody know there's a car GPS. I have my car, I have a GPS, but I never use it. Why I don't use it? Because they don't give you a road condition. I want to drive from my home to New York. They always tell me go to 78, but if there's traffic here, they cannot tell my GPS. But if I use my phone, like a Google map, it will tell you, okay, there's a traffic there, you may want to detour to reach your destination. It's a whole idea here. So the one we proposed, this DDM, is kind of the data guided GPS. The reason we call radar system is kind of like aviation. If you think about it, if you drive a pilot, you're a pilot, if you drive the jumbo jet from New York City to Beijing without ground controller, there's no way you can complete your task. You cannot drive from New York to Beijing without ground controller. So that radar system, just like a dashboard for you to look at how your data is going, how your trial is going. Now, who is the ground controller? What do we all propose here? As you know, the double-blind randomized trial and almost all the study, especially after phase 2 study, we have an independent data monitoring board. They can serve the role as the ground controller, as the ground controller in the aviation industry. So that is something I want to point out here. Now, this is just like a video I showed you, simulated data. As you can see, when we run this simulation, and the sample set is not good enough, we re-estimate and replot the boundary. And this study, simulated study, they eventually cross the boundary, you can success. I mean, the idea here is use the machine to learn what you observed, to update what the information you need, the effort you need later on. Now, as you probably see, there's some a lot of advantage by using an automation system, dynamic monitoring your ongoing study. I already highlighted something, mentioned something about sample re-estimation, population selection, even treatment selection. Also, safety is very important. You can do, and also more. So lots of advantage here. So I don't want to make too much, but bottom line here, I just want to point out what we are doing here is adaptation, dynamically adaptation. Simple as that. If you understand that adaptation, and also plus we have an automation system to dynamically monitor, apply the adaptation. That is the whole idea here. Okay. Now, what I did is also we did some like a simulation. This is the example how your system or method can improve the clinical trial for future. This is a simulation example. We kind of artificially assume. Okay, this is oncology study. The reason I say that because usually the average cost for per patient cost oncology is $100,000. This study, we estimate 30% treatment effect based on response rate. Then you need a 350 subject. Now, if you want to do the budget, is $35 million for this. Okay, now let's do this. I simulated this study, 10,000, same kind of setting. What if your drug doesn't work? It's the same as placebo. Now, the result tell you have 92% of chance to stop this study. And where you stop is about half of the total sample size here is 160 something. You stop in half. Now, how much money you can save? It's about $18 million based on my budget. Now, type 1 error is protected, as you can see here. Now, on the other situation, if your drug is much better than you expected, okay, you don't need that sample size. Then you also increase power. The reason you increase power is because the system will add more sample for you, but not too much. Okay, You also stop your study in some way, like more than half, 133 per arm. Now, how much money you save? You save up $12 million. So from this sense, this kind of tool will tremendously save huge money. But most important, I think, is the first role here. You can early detect a study which is hopeless drug or hopeless study to avoid financial loss and also ethically avoid the patient suffering. Okay. Now, this is a real example. I just gave you. This is a real example for the seizure for the kids when they have a seizure, and the kids will have a number of seizures per week counts. So we use this readout system to this independent data monitoring committee. They have a planned interim analysis of 50 because the total is 100 patients. And halfway they do the interim analysis, but because the enrollment ongoing, when the final, the real interim analysis happen is on the 52 patients. Now we plot this as the accumulated treatment effect and use the readout system. As you can tell, this is a very promising study. And also on here, we calculate, use the O'Brien framing type, the boundary. If you are like do the interim analysis at 52, not the 50. 50 is here, and 52, and the stopping boundary should be 2.928. Okay, now this study eventually they skip this second interim analysis. They also directly jump to by recommended by IDMC. So they don't need the second interim analysis. So they directly do the final, not directly. Eventually, they only do a final analysis for this study. Okay, so because of the time I remember when I was the independent statistician in this committee, one of the statistician, very famous statistician, who asked, based on this projection, this study, there's no need we can do a lot of interim analysis. So we keep going, and eventually this study announced complete successfully. Here is another example. Sorry. This is the example also we did this study. We spent 2 years for this study. There's no interim analysis planned. This is for the lung alcohol fatty liver disease. 2 years, then when we do the final analysis, we discovered the result is totally opposite. The study is a fail. Now, sponsor asked us, why is that? They don't believe, because based on the mechanism of the drug, they should be work, but this is not. So what we did is here, we reconstructed this study, retrospective. And the patient one by one, and also we create what we call the cumulative treatment effect, worst statistics here. As you can see here, this study, actually this drug is hurt the patient very early time. We don't know. Spend 2 years, and also this study should be stopped earlier at the beginning, but we don't know this. So this is a very extreme situation. So we presented this to the sponsor. They try to find out from the formulation, from the chemical. I mean, let's go back to the beginning to find out why that drug. Actually, it's a certain reason is because of this is cost the safety issue, safety concern, what we don't know. So this is the example I just want to highlight. Early discovery of this kind of situation is very, very important. Now, another example I just want to show you is the one for the very famous, the first remdesivir trial last year. Everybody should remember the last year what was happened in Wuhan, right? When the first this pandemic started. Gilead donated their drug, remdesivir, to Chinese government. Now, remdesivir is not originally designed for this disease because this virus is new to everybody. Now, in China, they also don't know this work a lot. So when they want to design a study, a 450 patient phase 3 study in Wuhan, they also have a traditional kind of like a adaptive design. They're going to re-estimate sample size in the middle way. Then the state of health of Chinese government didn't agree because they don't want wait until like end of the March to do the interim analysis to see what drug work. This drug work a lot. So their idea is think about if the drug work, they can have more people to use. If it's not work, they're going to stop early. So one of the person as statistician actually in the Peking University recommended use our system because I have a company back in China. So they contact our company in China. This is if you know Chinese, you know what this. Use our dynamic data monitoring for this study to monitor this trial. Now, this story I just want to tell you is interesting. Now, later on, I'm going to show you how this DDM make a discovery for this trial. As you recall, last year, about this time, April, okay? This is April 29, Chinese PI, they published this paper in NASA. And they discovered the remdesivir actually does not work. A little bit helpful, only 2-day improvement. Dr. Shi actually is an IDMC member for this trial. Maybe later on he can add some comments about this. This is 2-day improvement. On the same day, 29, if you remember these slides, if you watch TV, you remember this slide. On the same day, the morning this paper published, on the afternoon, Dr. Fauci make an announcement for the US trial. The super good, highly significant. They are going to stop the study in the middle way. This result also published. Okay, by the way, this background, you can tell where is that? It's the White House in Trump's office. Now, this is the US trial they published. There's over 1,000 patients. And also, the result is super significant at the p-value 0.001, very highly significant. Now, you may ask yourself, why? Why the same kind of population, same drug, why one study showing nothing? Another study is very significant. Now, based on this kind of question ourselves, we also come back to look at what's going on because we use our DDM to monitor this trial. We go back to see what's going on. This is the last DMC data. They published the data is based on this result. Okay. If you want to say the snapshot is this point. Okay. This is the result they published. But if you see whole picture, you probably have the different kind of understanding of what's going on. So you can see the 10 days, the remdesivir is doing very good, actually, starting beginning. On the 14th day, the remdesivir actually is doing very, very good. Okay, so and now, but unfortunately, when they get into this situation later on, the pandemic in China already got control. So no more patient. So when you see this data from here, number of subjects the same. The plots here is based on different date of the patient data. So this is 14 days data, same number of patient. This is 28 days. This is 21 days. Now, this discovery also think about we cannot say, oh, remdesivir trial in China doesn't work. Actually, it's work. Depends on how you look at. The reason because you are based on, if you take a look at this point, may not be a good result. So this trigger us to do a reanalysis for the China remdesivir data. We don't have the full data. We based on the published paper, they are using our plots here. They also use the what's called the response number, a percentage of the response. We reconstruct this and generate this kind of like analysis. This is the one. Okay, we have the DMC. We did analysis here. As you can see, there's a couple of differences between China trial and the US trial. First, China trial is the number one, the first trial in the for the COVID-19. And the WHO proposed some guidance, we call blueprint. The endpoint is time to clinical improvement. That clinical improvement definition is a 2-point improvement, 2-point. As you can see here, most of people is done a 3-point. 2-point means you almost recover. Okay, and also you see there's somebody should not get in. This is very mild, and the baseline score is 2, and they should get in. But unfortunately, this person get into the placebo group. So to prove improvement, to define the clinical improvement compared to in US is not based on the 2 scale. It's time to discharge or time to just 1 scale, in other words, and at least improve the 1 scale. And by the way, also US study changed the endpoint many times, 3 times, and also revised the sample size several times also. So now, when we reanalyze this data and we publish the result, and I think at least we correct some impression from public about remdesivir. And we think our conclusions remdesivir is still beneficial. So this is something for the China trial. I spent a little bit more time on them because that is a very interesting story and also demonstration about the dynamic data monitoring. So along this, we have several publications. One of them is regarding the DMC experience, which Dr. Shi is here. And later on, I'm gonna let him save some time, let him to give a comments. This is just a diary type of publication regarding the first, very famous first remdesivir trial. This is reanalysis of China COVID-19 trial. And also we published a paper recently, last month, regarding the DDM. This is kind of like a fundamental theory about this techniques we proposed here. Okay, so I'm almost finished my talk, and as you can see, I just want to make some comments about this. As you can see, the potential applications for this kind of technology. Although you may ask me, you may also criticize, say, okay, there's all a lot of the hurdle probably you're going to face. Yes, we agree on that. Regulatory environment, whether we can do this kind of fancy stuff in a clinical trial. The problem here is the solution is not going to change the traditional or classical study design. We provide a tool, just like the decision make when the aviation industry ground controller to make a decision and the sponsor is pilot to make a decision. We just provide a radar system for you. Okay, you see the whole picture. You make a more reliable decision. That's the whole point here. Now, we also provide other information like a trend, trending up, trending down. We use all those ideas like Wall Street is using every day, like average, moving average, all everything used on here. Because the calculation is not an issue. Technology is there. Now, the benefit from here, you can do all those like early termination for the hopeless study, population selection. Okay, you can do dose selection, signal, like a safety signal, many things, model checking, and many things you can do. Technology is not an issue. Now, another thing I just want to point out, because DASU is the organization very active. I'm so impressed for this organization and the young people. When I remember 30 years ago, when I was like their age, I always think about some new things. Now, technology is challenging us, how we're going to face for as a statistician for future clinical trial. Now, there are many things talking about machine learning, AI technology applied to clinical trial. I checked the publication, the area they are doing, apply the machine learning AI on this area, patient enrollment,
22.
and also trial compliance, quality control, and process efficiency. This is the area the current machine learning AI technology used for the clinical trial. None of them is try to apply to study design optimization. This is what we are doing, and also dynamic monitoring. I didn't say other thing for detect failure or success. So in other words, I want to point out there's a lot of things we can do. Still, we are in the early stage. So I just want to stop here and give some time to Dr. Shi, and also give some time for the questions. Thank you very much.
Thank you, Dr. Xie. Thank you so much for informative presentations. And I think we've got tons of questions. Okay, maybe we'll just start from the beginning. I think this is my advisor, Professor Yuan Ji from University of Chicago. He asks, if the patient population is assumed to be the same for the entire process of the trial, do we still expect to see a trend in treatment effect? I'm just a little confused on why there will be a trend if the patient population remains the same. If there is a shift in patient population, should the right approach be captured and accommodate the trend?
Yeah, let me try to, this is a very good question. Let me try to address this. And I wanted my co-author, Dr. Shi, and also Gordon Nance here also to address.
Sure.
If it's perfectly homogeneous and IID, the trend, what we're talking about is we do the conversion of the data you see. We use the p-value, which is under the Brownian motion. The assumption for the Brownian motion is you have a linear drift, right? Then that can be easier to tell. If you put this as a z-value, you may not easy to see that linearity. The trend is this, maybe you see up and down, but if the, what they call piecewise nonlinear, you can still treat as a linear in the situation, because if that is, but if you see the quadratic situation, which based on the assumption you talking about, homogeneity may not be the quadratic or other kind of like a type of trend. But if you see that trend, let's put it this way, if you see that trend, you may question about what's going on in your population. Because if you assume your population that way, you may not see that kind of situation. So the tool advantages here, if you see a very strange things, you may want to ask yourself what's going on here. So I don't know, Joe and Gordon, if you have anything to jump.
As you said, the graph that you plot in the beginning, it was a b-value, and in the radar screen, you can put in b-value or z-value, and those are the data points presenting the data. So for the Brownian motion one, you have under the alternative hypothesis, you always have I mean, if the Brownian motion structure is assumed, then you have a linear drift under the alternative hypothesis. Under the null, you will see a flat. There's no trimming effect. So in terms of the z-value, and then you will see a drift as well, but not in linear fashion. So the trend you're talking about is the drift parameter. And you can think intuitively, when you start, you always start with 0 point, and then you go up if there's a treatment effect, eventually you cross the 1.96 or other adjusted value. So you must go up. So there has to be a trend. Otherwise, you will stay at 0, right? 1 and it's 0. So there has to be a trend. I hope I answered the question to
Thank you, Joe.
Professor G. Okay.
Thank you.
Maybe
Okay.
next question.
Yeah, I think the next question is, could you clarify how the cost-saving decisions are made in the context of IRA-approved protocols?
I think it's a typo, it's IRB, right?
IRB.
Yeah.
Yep.
Well, I work in university, so I know how IRB works. They concentrated more on the ethical side. This is for data monitoring. I don't quite understand of the cost saving part. I think the saving can be saving lives, saving time, saving resources. I think the monitoring system will help to save lives and also save resources when the drugs are not working. When the drug is working, you also save lives by early stop. So IRB is not unfamiliar with data monitoring. Next question is.
Great. So the next question is, for DDM, is that real-time EDC data are typically not clean? Can you share some inputs on how to address this issue in practice?
Yeah, let me take that question. It is very good question. As you know, when the EDC directly stream the data into the DDM engine, data may not clean. However, if you really implement this like a system into your clinical trial, what we ask is we ask clean for the most interest of data you're going to monitor, which is primary endpoint, key secondary, or safety data. Those are part of data you wanted to as clean as possible. Just to keep in mind, the whole thing here, we provide a signal, not really to provide you some decision making. So may not be a very clean. For instance, if you see the data overwhelmingly good, although the data may not clean, the primary endpoint, this gives you a hint. You may want to think about a plan, a formal interim analysis, which require full-blown of data cleaning. Okay, so that's a good question. Now, a lot of things, the application for this, we have this DDM engine as integrated with the EDC system. And also we have our called API. So API will be either communicate with other database or even upload the source data sets. So we did actually for several IDMC, our independent statistician, just upload source Atom datasets into the DDM. Then automatically we can plot these things. You don't need to have to directly communicate with a system. Now, communicate with system, you can monitor your data kind of like a live kind of situation. But the upload is kind of hybrid situation. So both way will be okay. Next question.
Great. So next question is also from a statistician from the industry, and he has two questions. The first question is, how do you control for multiplicity? And the second question is, just curious, how does the DDM work in practice? How early should you start monitoring? And as for the sample size re-estimation, is it possible that one point the system tells you to increase the sample size, and then at the next time point, we see a better effect? Thus, there's no need to increase the sample size, and we need to override a previous decision.
This is very good question, actually. Now let me try to answer the second question first. Joe probably jump into first question. Second question is about the, this is talking about the where you do sample size, where you pick up, how comfortable you want to do that kind of sample review. What we did here, I mean, in this region, purple region, the sample size ratio is, we continuously kind of display as more patients get in. The ratio is based on your previous sample size estimate. For instance, the ratio is greater than 1 means you increase, right? Greater than, less than 1 means you decrease. That's also fluctuating up and down. So we need to find a region where you feel comfortable. So if that data is consistently in this middle region, you're probably thinking about to do the sample size estimate, re-estimate. Now, where to start it? You start the monitoring based on our simulation. Too early is not good. The data is very fluctuated. You can, the real data I just showed for other example, the early is very fluctuated, later on stable. We recommended after at least 30% of patient to start your monitoring. Okay, so that is relatively stable. We did some simulation regarding where you can start monitoring. By the way, in our DDM system, you can move. I plotted this to a vertical line. Actually, they're starting the monitoring. You can move this. If you want to start it to monitoring, you can move to the earlier time or later time, depends on you. For the first multiplicity, Joe, can you answer that question?
Okay, I think this also will answer other people. I see the questions coming from. Many people have this controlling type 1 error thing here. We in the DDM is a inclusive system. By inclusive, I mean you have start from the group sequential, the classical group sequential, and that's the first option you can specify. You can specify the number of interim analysis and plus one final. And you can have a fixed schedule, DMC meetings. So you have information time and cutting the time and follow through that. You pre-specify your land-based spending function. There's a family of them. So other software packages have all implemented a classical group sequential. And they're very flexible and with possible changes on the schedule and the number of analysis. But it is not equipped to change the maximum information. Therefore, the second option is so-called adaptive group sequential as opposed to the classical group sequential. So adaptive group sequential will be the next level. All the classical group sequential you can do in the DDM. But in addition, for the adaptive group sequential, you have two more features. One is the sample size re-estimation. You can increase the original maximum information if necessary. And also you can do futility analysis, blinding or non-blind futility. But all the interim analysis are on a snapshot, so not on the cumulative trend So this is adaptive group sequential. You use a single point snapshot to estimate your trimming effect. And there's no auto warning system until scheduled DMC review meeting, then you do a snapshot there. Okay, so you go to the deepest level, which is the DDM. All the adaptive and classical group sequential are implemented there. And the new features are the data availability. So at any time when the data is ready, so whatever that the clean stage that you feel comfortable, anytime the data is ready in the DDM system, then you can start this. So good feature is data availability at any time when data is ready and the system is ready. Okay, and then is the next one is the data availability in a cumulative trend analysis way, not by a snapshot, because we know that the conditional power, it depends on your trimming effect, but we don't know trimming effect. And snapshot give you, based on the Brownian motion theory, there is no memory needed there. However, as Dr. Shea pointed out, that the data may not be the Brownian motion, and may be modified, what I'll call modified or fractional Brownian motion, and then the trend analysis displayed by this DDM can be utilized. So you're not dependent on a single shot there. And for the multiplicity part, as I said, is data ready anytime, and it doesn't mean you just every night, whenever you have time, or when there's new information, you will turn on the DDM. You still follow your scheduled analysis. So, for example, you can do. Let me do use an what you watch cable TV. You can prescribe by pay-per-view or pay-per-plan, right? And pay-per-view is each time you look at it, you watch a program, you can pay your price. So the price here is your alpha level. And you can also pre-specify a pay-per-plan if you turn on the DDM option, and so that's where it is different from the other software available. But in terms of the theory preserving the type 1 error is all well established in Lindeman's paper. If you do a pre-preview, you start with a specified in the classical group sequential. You also can change your frequency and your timing, your schedule as well. For pay-per-plan, for example, you can be silly. You can do, say, I want to do 10 or 20. Nobody is going to monitor a trial with 20 equally spaced interval, but you can do it, okay? You can pay per plan. You just pre-specify a 20 equally spaced interim analysis. You can even push more limit if you want to. So all those theories are already established. So the alpha type 1 error is preserved. There is no something new to DDM in preserving alpha. Then the adaptive group sequential, even including sample size re-estimation. The adjustment of critical value or the test statistic to control type 1 error is all published. And this also answers another person's, Dr. Porto's question about the publication. We do have a publication of DDM, which mentioned this, and also given an example where you use a trend to estimate trimming effect by piecewise linear trend, not a Brownian motion. You can use a weighted slope to estimate the trimming effect for your conditional power calculation. And that's more robust to the one snapshot estimate of the trimming effect. And that's an option in the DDM as well. I hope I answered the question. Can you hear me?
Yeah, thank you,
Okay.
Professor Shi. Thank you, Dr. Xie. I think we're about time. And we will collect all the questions in the chat box and post Dr. Xie and Professor Shi's response on the YouTube comment sections of today's webinar. And I just want to thank you again, Dr. Xie and Professor Shi, for today's great presentation. And thank you everyone for attending our session today.
Thank you. Thank you, everybody.
Thank you, everyone.
Yeah,
Bye.
thank you very much. Yeah, appreciate it. Have a good weekend.